Great. Okay, so let's get started then. We are very privileged to have Professor Sir Munir Pirmohamed with us today, especially in this incredibly busy period for him. He is the David Weatherall Chair in Medicine at the University of Liverpool, a consultant physician at the Royal Liverpool University Hospital, and directs the MRC Centre for Drug Safety Sciences, the Wolfson Centre for Personalised Medicine, and HDR North. He is also an inaugural NIHR Senior Investigator, a Fellow of the Academy of Medical Sciences, a Commissioner on Human Medicines, and a Non-Executive Director of NHS England, as well as the President of the British Pharmacological Society. So he chairs the Commission on Human Medicines that advised the MHRA on the vaccine that was approved earlier today, and he'll be taking some questions on that at the end of the talk. But the focus of our lecture today will be on pharmacogenetics. And so with that, I'll hand over to you, Sir Munir.
Thank you very much for the invitation. Let me just share my screen. Okay, can you see full screen?
Yeah, all looks good.
Brilliant, thank you very much. Okay, well, thank you very much for the invitation. I was asked to do a talk on pharmacogenetics, past, present, and future. And I'll focus in on that, and I'm happy to take questions at the end. So why is pharmacogenetics important? So I thought I'd give you a perspective from the National Health Service. I work in the National Health Service. I'm a non-exec director for NHS England. And if you look at what we do with medicines, the UK NHS deals with over 1 million patients every 36 hours. It is the sort of, largest integrated healthcare system in the world. We prescribe 1.1 billion drugs in the community, and the cost to the NHS is about £18 billion per annum. Unfortunately, that leads to a lot of issues given the scale of this. There are quite a few medication errors, as you can see, and there's quite a lot of medicines wastage going on. Medicines wastage because patients don't take medicines as intended, so they just throw that away, and sometimes medicines wastage because of the side effects and so on that happen because of the medicines and the way we use them. And the problem that we face in any healthcare system is there's quite a lot of variability in drug response. And so what I mean by that is that there's variability in drug efficacy. Clearly, when I, as a clinician in the NHS, when I prescribe a drug, I prescribe it to have an efficacious effect, the benefits. And I try to minimize the risk of it, minimize the toxicity or side effects. But unfortunately, at the moment, I can't predict the first time I prescribe a drug, the patient will respond to it in a favorable way. And it's been shown that the vast majority of drugs that we have in the British National Formulary and in the American equivalent, more than 90% only work in 30 to 50% of people. So with some drugs, such as antidepressants, we have to give the drug, wait for 3 months to see whether the patient's improved. They haven't, then we have to try another drug, and then we have to try another drug. So some patients may go 9 months with 3 different drugs and may not improve at all. So that's a waste for the, obviously in terms of drugs, but also really bad for the patient in terms of outcomes because of this trial and error approach. On the other side is the sort of issue of adverse drug reactions, the side effects associated with drugs. Many of those are serious enough to cause hospital admissions, and we've undertaken some of the largest studies in the world. We've shown that 6.5% of all hospital admissions are due to adverse drug reactions. When patients are admitted to hospital because of a particular disease process, the prescriptions are being changed all the time in hospital because of the changing physiology, pathology, and 15% of those patients will develop adverse drug reactions in hospital. And if you take those two figures together, 6.5 and 15%, and extrapolate that to the whole NHS bed base, at this very moment in time, there are 8,000 NHS beds occupied by patients with adverse drug reactions. And if you consider that each NHS hospital has about 800 beds, that's 10 800-bed hospitals occupied by patients with adverse drug reaction at a cost to the NHS of at least 1 billion, and that's probably a conservative estimate. So that's a huge cost. Issues with adverse drug reactions are important not only for the NHS, they're also important for industry. We now know that to develop a new drug costs well in excess of $1 billion. And if you look at this paper, which is from Oxford, from Jeff Aronson, he looked at numbers of drugs which were withdrawn between 1953 and 2013. And what he found was that 462 drugs had been withdrawn. Most of it was related to anecdotal reports, i.e. case reports and so on, rather than randomized controlled trials. And they affected many different organ systems, such as a liver, immune-related reactions, and the heart. And what they found was that the median interval between the first ADR report and withdrawal was about 6 years. But if you look at this graph, it's very interesting that in 1750, you can see this was the launch year. It took 200 years between launch and withdrawal of a drug. But fortunately, in this century, we're getting better, 6 years. But we need to get much better still in terms of trying to identify when drugs come onto the market and cause problems and when they need to be withdrawn. Better would be to make sure the drugs are actually safe and don't need to be withdrawn. And pharmacogenomics may also play a role in there. So what is it that determines variation in drug response? Well, you can divide it into two broad categories. The first one is pharmacogenetics, what the body does to the drug, absorption, distribution, metabolism, excretion. And then there's pharmacodynamics, which is what the drug does to the body. Now, in pharmacodynamics, basically drugs will interact with molecules within the body, and their typical ones, which are drug targets, are ion channels, receptors, enzymes, and nucleic acid. And the factors which determine variability are many different factors, including, for example, multiple drugs. Sometimes gender can be a factor, other diseases can be a factor in terms of determining variability in response. But there are also genetic factors. We do not know overall what the genetic contribution is to variation in drug response, but it will vary from drug to drug. Some cases it may be almost 90 to 100%, in other cases it may be less than 10%. So at the moment, when I prescribe a drug to a patient, I will take into account some of those factors, such as the age of the patient, such as gender, and other diseases they may have. For example, if they have renal failure, chronic kidney disease. So, and the British National Formulary again has instructions that if you have somebody who has got kidney failure or impaired kidney function, then you should be reducing their dose of the particular drug you're going to prescribe. So here's a beta-lactam antibiotic, and it says that you should reduce the dosage at these kind of creatinine clearances. And these kind of dose instructions are based on pharmacokinetic, pharmacodynamic modeling studies. They're not based on randomized controlled trials, because you would be impractical to actually undertake randomized controlled trials in this situation. Now, when we have a genetic variant, we completely ignore that. And the genetic variant may have exactly the same effect as renal failure, but at the moment it is completely ignored in most cases. And what we're trying to say is that actually, now that the technologies have improved, we need to be able to introduce genetic variation in terms of drug dose and drug choice, so that that can be implemented into clinical practice. So I'm going to take you from past, present, to what I think will be happening in the future. So pharmacogenetics is not a new area. It has been around since at least 1957. This is a German pharmacologist called Friedrich Vogel. And he coined the term pharmacogenetics. He gave it a different definition, but the International Conference on Harmonization in November 2007 called it the study of variation in DNA sequence as related to drug response. In 1997, the editor of Nature Biotechnology, Marshall, coined the term pharmacogenomics. And since we were able to look at the whole genome this really relates to the fact that we don't need to look at individual genes, but now looking at the whole genomes and RNA as well in terms of determining drug response. And these terms are used interchangeably, but the ultimate aim is to improve drug response, reduce variability of drug response. In fact, pharmacogenetics is much older than 1957. The first proponent probably, although he did not know it, was Pythagoras. And he wrote many things, but one of the things he wrote was be far from fava beans consumption. And he held that belief so strongly that he met his death in ancient Italy in Cretonia because he refused to cross a field of beans. At least that's what the story is on the internet. And there are many theories why he held those particular views about fava beans, maybe because it contained the souls of our forefathers, or maybe because they look like testicles. A medical reason may be that eating fava beans causes quite a lot of flatulence. But probably actually the medical reason why he did have that view about fava beans is that it causes a problem called favism. And favism is well known to occur in certain Mediterranean populations, largely because of a particular genetic issue that they have, and I'll come back to that. And what happens with favism is that you can get red blood cell hemolysis. Your red blood cells just burst open, hemoglobin pours out of the red blood cells and goes into your renal tubules, block the renal tubules, and causes renal failure. So it can be quite a serious, life-threatening adverse reaction. We now know that this is related to a deficiency of glucose 6-phosphate dehydrogenase. And this is the most common human enzyme deficiency in the world. So what has that got to do with drugs? Well, actually, there are drugs where we have to check for G6PD deficiency before we give the drugs. So when I was a junior doctor, the first time I did a pharmacogenetic test was when a woman came in to the hospital I was working in with Vivax malaria. And you can see that on the slide here. And the treatment for that is primaquine. And so I thought, well, if I'm going to give her primaquine, I need to rush to the hematology lab, get her G6PD measured, and then determine whether I need to give her the full dose or half the dose of primaquine. Fortunately, she was negative. I was able to give her primaquine. She responded very well, and she was able to go home in a few days. But if I hadn't tested it and she was G6PD deficient, I would have caused hemolysis, which would have had no impact on her malaria in terms of cure, but it would have made her much more sicker because of the red blood cell hemolysis. Now, if you look at the history of pharmacogenetics after, even before 1957, some people were talking about it at that time. For example, Archibald Garrett in 1910 published a book called Garrett's Inborn Errors of Metabolism, and he was basically talking about some pharmacogenetic defects, although he didn't coin the term. Later on, Werner Callow, published a book called Pharmacogenetics, one of the first textbooks on heredity and response to drugs. And Werner Callow had to flee from Germany during the war and went to Canada and did all his work there and was one of the pioneers of pharmacogenetics. Then David Price Evans took the mantle looking at NASTAR transferase, particularly looking at anti-TB drugs, and he was professor of medicine at Liverpool and did quite a lot of work between Liverpool and Johns Hopkins in the United States. Now, if we look at how we actually assess for pharmacogenetic variants, in the 1960s and 70s, it was based on phenotypic testing. That is where you give a probe substance. The subject will take that probe substance, and then you collect urine for 6 hours. And in that 6 hours, then in the urine, then you can look for breakdown products. And depending on the ratio of those breakdown products, you can determine whether the patient has a variability in that particular enzyme. In the 1990s, with PCR becoming available, people started looking at single gene defects in terms of pharmacogenetics. And then obviously we've gone to multiple genes, genome-wide association studies, and now it's very easy to be able to take whole genome sequencing approaches as well. And clearly, one of the major advances ever in terms of science is the completion of the human genome. First draft in 2001, second draft in 2003, and that's been enormously valuable in terms of the data that's been made available, online, free, so for scientists all over the world to be able to look at that and really pursue many different areas, not just pharmacogenetics, rare diseases, cancer, etc. We know from all the work that's been undertaken that 99.9% of the human genome is exactly the same, irrespective of where you live in the world, but 0.1% is different. 0.1% doesn't seem very much, but given that there are 3 billion base pairs in the human genome, then 0.1% equates to 3 million, and 3 million variants, 3 million differences are likely to lead to a lot of variation in the human population, and that's important for evolution as well. But it determines many different characteristics, including variation in drug response. And I'm always amazed by how big the human genome is, and this illustrates it, in terms of how many books the whole human genome occupies. 130 books, and if you were to read every letter in the human genome, it will take you 100 years and so on. So I think that's an amazing fact. The human genome is a fascinating area to work in. Now, if I bring you to the present day, given that we've got so much information now on the human genome, people started looking at how many people carry pharmacogenomic variants, and is this really important to healthcare? And this is from the US Veterans Program in the United States, and where they looked at 7.7 million US veterans, and what they were able to show was that 99% carry at least one actionable variant in a pharmacogene. So the majority of us, almost all of us, carry pharmacogenomic variants. And 2.9 million, i.e. 37% of the 7.7 million, has started treatment with at least one level A drug. And what level A was a gene-drug interaction where there's a high level of evidence from well-designed, well-conducted studies that you really need to be able to consider either changing the dose or changing the drug that you're going to give. So it highlights that pharmacogenomic variants are common, and we are often using drugs which require pharmacogenomic variation. And this is a paper from Nature, which looked at FDA-approved medicines, about 1,200, and found that 93% were affected, 7% overall were affected by actionable pharmacogenes. And that equates to about 18% of the prescriptions in the United States. So it's a huge issue that we're really not looking at all in any great detail at the moment. So we've been working in this area and trying to develop the evidence base, which allows us to be able to then take this into clinical practice. I'm going to show you some exemplars of some of the work that we've done, plus also some of the work other people have done. And I'm going to start off with one particular enzyme that you may have heard about, a cytochrome P450 enzyme called cytochrome P450-2D6. It's been known about since the 1970s. And we know that it's very complex in terms of its genetic architecture. 5 to 10% of individuals are called poor metabolizers because they lack, they have genetic variants, which causes lack of the enzyme. About 10 to 15% are intermediate metabolizers because they may be heterozygotes. And then there are a small number which are ultra-rapid metabolizers, which means that they have more than 2 copies of the gene. And the majority of families have 3 copies of the gene. There are some families which have 13 copies of the gene. So if you've got 13 copies of cytochrome P450-2D6 and you take a drug which is metabolized by cytochrome P450-2D6, you'll have a lack of response because you're going to be over-metabolizing that particular drug. And if you look at the British National Formula, again, 25% of drugs are metabolized by this particular enzyme, but 8% of the UK population lack the enzyme, and 2% of the UK population have more than 2 copies of the enzyme. And at the moment, we completely ignore the variation in this enzyme when we're prescribing drugs. But hopefully that is beginning to change. So here's an example of a drug which has been come onto the market not long ago called eliglustat. It's used for Gaucher's disease, which is a rare disease, which is a lysosomal storage disorder. And when this drug was being developed, they were able to identify the drug is metabolized by cytochrome P452D6. They were able to undertake pharmacogenetic studies, which showed that really, if you are an extensive metabolizer or intermediate metabolizer, you need 84 milligrams orally twice daily. But if you're a poor metabolizer, you should halve the dose. So this is now a common practice for this particular drug. But there are many other more commonly used drugs which we completely ignore at the moment in terms of pharmacogenetics for cytochrome P452D6, and codeine is one of those examples. Codeine is a prodrug. It's broken down into morphine to provide pain relief. You need cytochrome P452D6 to break down codeine into morphine. So if you lack cytochrome P452D6, what happens is that you can't actually form any morphine, so you don't get analgesic relief. So at least 1 in 10 people in this country who are given codeine will get no analgesic benefit because they can't break down codeine to morphine. And some people may say, well, this is the drug's not working in you because you're just a wimp. In fact, that is not correct. They may have a biological basis for why the drug is not actually working in them. If you look at the other aspect of cytochrome P452D6 and codeine, if you look at the ultra-rapid metabolizers, that is people who have more than 2 copies of the gene, it can actually cause toxicity. So here's a case report of a mother who was given codeine because of pain after labor because she'd had an episiotomy. What was unknown at that time was the mother had duplication of her cytochrome P452D6, 3 copies of the gene, and there she was an ultra-rapid 2D6 metabolizer. And she was breastfeeding her child, and the child started becoming unwell, really, had difficulty in breastfeeding, became lethargic, and unfortunately was found dead on day 13. And when they started investigating that, they found that the morphine concentrations in the breast milk were grossly increased, much more than you would expect if you had 2 copies of the gene or 1 copy of the gene. And now the drug label for codeine actually warns against using codeine in breastfeeding mothers because of the risk of this. A second example is looking at warfarin. Now, warfarin is a very commonly used anticoagulant. About 1% of the UK population use warfarin. It is used for deep venous thrombosis, pulmonary embolism, for atrial fibrillation to prevent clots. Unfortunately, with warfarin, we have no idea what the dose will be in the patient when you start, because patient dosing can vary from 0.5 to 20 milligrams. So it's trial and error to get them to the right dose. So there's 40-fold variability in dosage. And if you get it wrong, what happens, because warfarin is anticoagulant, you can get bleeding, as in this 82-year-old woman who had a calf hematoma. So what we've been interested in is to find out how we can actually improve the dosing of warfarin. And if you look at the pharmacology of warfarin, warfarin is metabolized by cytochrome P452C9. And there are genetic variants of cytochrome P452C9. And one would expect that if you have a low activity variant of cytochrome P452C9, you'll metabolize warfarin to a lesser extent, and therefore you need a lower dose of warfarin. Warfarin inhibits an enzyme called vitamin K epoxide reductase complex, which affects the vitamin K cycle and therefore prevents the formation of vitamin K dependent clotting factors 2, 7, 9, and 10. Again, this is variably expressed in the population, and therefore you'd expect that individuals with low activity variants would require a lower dose of warfarin. And we undertook a genome-wide study in 714 people recruited from the UK. And this is a Manhattan plot with all the chromosomes on the x-axis and the minus log 10 p-value. Because we're looking at a whole genome association study where you do 1 million different variants on each individual, you have to correct for multiple testing, and so the p-value that is said to be significant here is 5 times 10 to the minus 8. So you've got to be below 5 times 10 to the minus 8 to be significant. And you can see we have 2 peaks here, one at 10 to the minus 30, highly significant, the cytochrome P452C9, the enzyme that breaks down warfarin, and also with the vitamin K epoxide redox complex with a p-value of 10 to the minus 50. And when we look at these two factors account for about 40% of the variation in daily dose requirement. There are other factors as well, age. So age accounts for 11%. As a patient gets older, you require less warfarin because liver mass gets smaller. As one gets heavier, you require less warfarin. As you get heavier, you require more warfarin. And that needs to be taken into account. Interacting medications are also important as well. So when you have all these factors, we can actually account for about 60% of dose variation. So what we did was to combine these factors and create an algorithm. And when you have an algorithm, you then want to show the utility of the algorithm, whether it will work in patients. So we then undertook a randomized controlled trial, and we'll publish this in the New England Journal of Medicine. The randomized controlled trial was undertaken in the UK and Sweden. And what we did was to randomize patients to get standard dosing, which is what we do in the UK at the moment, compared to genotype guided dosing, which is where you can actually identify what the genotype is and then dose according to genotype. And we identified the genotype using a point of care test. Basically, we take a small amount of blood, put it into that machine you can see on the screen, and that gives us a result on 3 variants in those 2 genes I've just shown you within 2 hours. And then we can put the genetic information into a web-based dosing algorithm, and that tells us what the dose should be in a patient who's had genotype-guided dosing. So what we found was in the control arm, which is standard dosing arm, the time in therapeutic range, which was the primary outcome measure, was about 60%. And that's pretty good for 3 months. Normally, you know, in normal clinical practice, you'd probably be getting about 50% time in therapeutic range, but in a randomized control trial, we were able to get 60%. But despite this very good control arm, we actually beat it in terms of genotype arm with 67.4% time in therapeutic range, which was highly statistically significant. So this was a randomized control trial. The next thing that happens when you do something like a randomized control trial is people say, well, it's protocol driven, it won't work in the real world. So we then went on to do a real world study where we implemented this in 3 clinics in Northwest England. What we did was to refine the point of care genotyping test. So you could now get the genotype within 45 minutes instead of 2 hours. We then trained nurses to be able to do the genotyping test and left the genotyping platform with the nurses in the clinic. They were able to do a mouse swab, put it into the machine, get the results in 45 minutes, and then dose according to genotype. And just to remind you what we got in a randomized control trial, a 7.1% difference. In our implementation study with nurses doing it by themselves in clinic, we got almost exactly the same result, a difference of 7.5%. Again, showing that you can be implemented in a real-world clinical setting. As this was an implementation study, we asked patients and nurses what they felt about the genetic dosing. And there were no adverse comments, really, apart from the fact that most nurses, 8 out of 13, felt the genotype-guided dosing did not fit in well with the running of the clinic, i.e. we disrupted normal clinical pathway. So normally when a patient comes into clinic, they're seen by the nurse. You have your INR test, you have your blood test result available, and you then tell them what the dose should be, and they go away, and it takes 10 minutes. But here the patient was coming in, they were requiring a genetic test, which took 45 minutes. So they had to wait for not only the first 10 minutes, but also another 45 minutes. And then they had to come back for another 10 minutes. So overall, the clinic time went from 10 minutes to an hour and 5 minutes, and that disrupted the normal clinical pathway. So we need to think about how we can improve that clinical pathway and therefore help with the implementation of technologies such as this. Now, it's now widely accepted that pharmacogenetics is important for warfarin, and there are now clinical guidelines which have been developed, and it is being used in many different clinics in the world, particularly in the United States. And the last example I want to show you is about a serious adverse drug reaction. And the picture here is of a patient with something called toxic epidermal necrolysis, where your skin sloughs off after 2 weeks of the dose of a drug. and it is rare, fortunately. It occurs in about 1 in 100,000 to 1 in a million patients, depending which drug you're looking at. It has a mortality of about 30% when patients develop this kind of reaction. And there's been a lot of interest as to why this may be occurring. This is an immune-mediated reaction, and people have focused on the immune system and the HLA complex, which is on the short arm of chromosome 6. And HLA can be divided into class 1, HLA A, B, C, class 2, DR, DQ, and DP, and in between class 1 and class 2 is the HLA class 3 region, which codes for genes such as TNF alpha and so on. So work we've undertaken, plus many other people have undertaken, has shown that there are certain HLA alleles which predispose to serious adverse drug reactions. And the best example of that comes from a drug called abacavir. This is a drug which has been used in HIV. It is very effective, but unfortunately about 5 to 7% of individuals get a serious reaction affecting the skin, lung, which can be fatal in these patients. And work which was undertaken in Australia and then North America and then by ourselves were able to show a very strong association with HLA-B star 5701. And people then went on to do a randomized control trial where they were genotyping people with HLA-B 5701 before they put the patients on abacavir. If they were positive for HLA-B 5701, the patients were then put on a different drug and not on abacavir. And this was highly successful as a randomized control trial in terms of showing that there's utility in using HLA-B 5701 typing. We actually identified this in 2004. We were able to do cost-effective analysis showing that it would be cost-effective for the NHS to be able to implement this into NHS clinics. And this data was used by HIV clinics in the NHS. And by 2006, every patient in the NHS was getting 5701 genotyping before they went on abacavir. And the net effect of that has been dramatic. Before genotyping for this particular allele, the incidence of abacavir hypersensitivity was 5%. It is now less than 1%. And I haven't seen a case of abacavir hypersensitivity for the last 7 years or so. Now, if you look at the literature in this area, there have been many other studies which have shown very strong associations between different HLA alleles and different types of drugs and different types of reactions. And these are the kind of different kinds of designs people have used. These is a list of some of those associations which are really strong enough at the moment to be able to think about implementing them into clinical practice. And what we've done is developed an HLA panel, a multi-allelic panel, which can now be made available to the NHS and other healthcare systems for much cheaper than conventional single locus tests which are done in the NHS at the moment. Our panel can also be available in terms of turnaround time within 48 hours, which is really important for the physician to be able to make a change to the medication or decide which particular medication should be given to a patient depending on what the HLA allele shows. So just to move on, I think we face major issues as we go forward over the next 10 to 15 years. We have an increasingly elderly population, as have many other countries, including the sort of Southeast Asian countries such as China and Japan as well. As one gets older, one has a deterioration, renal function, liver function, respiratory function, cognitive function, mobility. This is a time when people are getting multiple diseases affecting multiple organ systems. And this is time we often give patients multiple drugs. And I frequently see patients who are elderly with 10, 15 drugs that they're taking every day. And you may have relatives who take this many drugs. Unfortunately, these patients are never tested in clinical trials. You know, we are taking data from relatively healthy younger individuals, 40, 50-year-olds, and extrapolating them to our 80-year-old patients who are on multiple drugs. And often we don't know, how they're going to react to this in terms of the side effects that they're going to get and the positive benefits they're going to get as well. So in order to try to tackle that, what we're trying at the moment is carrying out a randomized control trial, which falls under this particular program called Ubiquitous Pharmacogenomics. It's funded by the EU, 15 million euros. And the idea here is to implement preemptive pharmacogenetic testing in real-world clinical settings across 7 EU sites. Liverpool is the only UK site, but you can see this other 6 other countries that are involved in undertaking the trial. And what we've been doing, it's been running since 2016, and the first 18 months or so was really to sort out the infrastructure for undertaking the trials. So we basically developed the information technology tools we required for undertaking the trial. We then put in the necessary genotyping platforms in laboratories across the whole of Europe. We developed the guidelines in terms of what to do when you have a genetic variance, and then took training education of all the people who are going to be involved in the trial. And what we've done is recruited 7,000 individuals to the trial in a cluster randomized design. And what our outcome measure is to determine whether this reduces adverse drug reactions by having pharmacogenomic choice of drug or dose. And the data is being analyzed at the moment and hopefully will be reported next year. In terms of the panel that we developed for this particular trial, this panel has 12 genes in there, which includes 44 variants in those 12 genes and covers 39 drugs which are used widely within most healthcare systems in the EU. When patients have had this genetic test, we give them a card with a QR code, and the QR code can be linked via the phone to an internet site which tells the physician as to what particular variants they have, so they can the physician can then make a change to the particular drug therapy they're going to give. So this will hopefully read out next year, and with the results to see whether it actually makes a difference clinically. So obviously, many of you will have heard of the 100,000 Genomes Project, which has been going on, first announced at the time of London Olympics, completed in end of 2018 with 100,000 genomes, and in late 2019, these are the numbers of genomes which have been completed and results which have been sent back to patients, including for rare diseases, 20 to 25% actionable findings for rare diseases, and 50% of cancer cases contain potential for therapy or a trial in the 100,000 Genomes Project. So what the whole genome sequencing is now becoming mainstream. It is being introduced into the NHS. I can, if I needed a whole genome sequence done on a patient with a particular condition, I could refer it to a particular laboratory, which would then be able to do this whole genome sequencing. But what we're finding is that we get whole genome sequencing is that we're getting a lot more data, and it is sometimes difficult to be able to interpret that data because we do not know the function of those variants that we identify, particularly the rare variants. And the rare variants may have a substantial effect size in terms of predisposing to disease or predisposing to variation in drug response. And so we have got a lot of work to do for the future, and I'm heading towards the future in terms of what we need to do, really look at, so that we can actually take advantage of the knowledge we have in the human genome to be able to extrapolate that to making drug therapy much better for our patients. And in order to start to work on this and really try to improve things, so the 100,000 Genomes Project is undertaking a pilot project at the moment. And really the questions the 100,000 Genomes Project is asking with regard to pharmacogenomics is, if you have a particular pharmacogenomic variant, what variant should you use in order to inform the clinician? What do you do with rare variants? And by rare variants, I mean that they occur at a frequency of less than 1% of the population. How will you make sure that the genomics data is embedded in the patient's electronic healthcare record, so that as they get older and they started on more and more drugs, that the clinician can have the information at the tip of their fingers, so they can actually make the change in terms of drug to be used? And how will doctors be able to interpret the results? Because most doctors have been trained in pharmacogenomics and take action. And so we are looking at a particular gene called dihydropyrimidine dehydrogenase, DPYD, which metabolizes anti-cancer drugs such as 5-fluorouracil, and these 4 variants initially are being looked at as a pilot in this particular project. Now, if you look at the 100,000 Genomes Project, and this is data from the 100,000 Genomes Project, we find that the majority of variation in the 100,000 Genome Project is actually rare. Only basically about 2.6% of single nucleotide variants and 5% of indels have a minor allele frequency of greater than 1%. The majority of the variation is actually, in fact, is extremely rare, less than 0.1%. And we will need to deal with this as we move forward. How do we actually take into account the rare variation in terms of improving drug response and improving diagnosis of disease? And so really, we're heading towards an era where we want to implement pharmacogenomics into healthcare systems. And if you look at what's happening globally, there are certain sentinel hospitals in the United States, for example, the Mayo Clinic, Vanderbilt, etc., are starting to think about implementing pharmacogenomics, and some other hospitals, for example, the Netherlands and so on. However, most of these are focused on individual hospitals. Qatar is looking at the possibility of doing a whole system implementation of pharmacogenomics. But the NHS is the largest healthcare system, integrated healthcare system in the world. And therefore, it is really important to determine whether we can actually start implementing pharmacogenomics into the NHS. And in order to do that, I've been working with the NHS England, plus other people have been working with NHS England to see how we can think about undertaking this. We held a workshop, first of all, in early 2018, and then identified potential candidate gene pairs that were important for taking into pharmacogenomics in the future. And what we then did was to develop a scoring system for pharmacogenomics to identify which particular genes we want to take forward. And this was dependent on the allele frequency, the level of clinical benefit, how frequently the drug was used in the NHS, and also the level of evidence, how good is the evidence that a pharmacogenomic variant will affect drug response. We also had to take into account technical considerations, how would you genotype for that particular variant. And the list, gene drug pair list, is being drawn up by the NHS to be able to incorporate into the genetic testing directory. And hopefully next year at some point, the physicians may be able to order which particular pharmacogenetic tests, depending on the drug they're putting the patient on. And this will depend on several factors in terms of genetic testing directory, the cost effectiveness. Is it cost effective to be able to implement that into the NHS? How can we actually undertake some of the genotyping? Is it possible to do by sequencing? Is it possible to do by genotyping arrays? How are we going to make sure that the genetics labs in the NHS are not overwhelmed by lots of requests here? So we've created eligibility criteria. This is decision support tools is not being sorted out yet, and I want to come back to that in a few minutes. And obviously, how do you validate and report to the clinician that the patient has a variant in their genome, which may be important for their drug prescription? We've published on this recently, what we discussed in the workshop, and what the key barriers, opportunities, and challenges are. And I won't go through this table, but it's available online for anybody who wants to read it. One of the things that's become evident from work that we've been undertaking, plus a lot of the work that's going on all around the world, is majority of studies which have been undertaken in genomics as a whole and pharmacogenomics as well have been performed in individuals of Northern European ancestry. And the genomic findings in white populations do not necessarily map across to Black populations and to other populations, Indian, Chinese, etc. So therefore, genetic tests which is developed using data from white populations will not necessarily be useful in Indian, Chinese, or African patients. So it is really important that we actually improve work we undertake in different ethnic backgrounds, so that when we develop genetic tests for the future, they actually are there for the whole global population rather than exacerbating race inequalities. Another major aspect that we need to consider for pharmacogenomics implementation for the future is really about education and training. Most people do not get much pharmacogenomic training in their curricula. This includes medical students, nursing students, pharmacy students, etc. And so the public also have very little understanding of pharmacogenomics, and clearly that also needs to be improved. And there are many challenges in undertaking that pharmacogenomics education. Important amongst, if you look at your crowded curricula, then where's the space where we're going to introduce a whole new curriculum on pharmacogenomics in there, and that needs to be looked at in many different areas and many different professions. So just to finish off, really with a couple of slides. At the moment, when we are implementing something into clinical practice, we have this evidence hierarchy, which says that if you have a randomized controlled trial, that's the best evidence, and you can use that evidence to be able to change practice. But if you have case reports, ideas, opinions, that's at the bottom of the pyramid, and it's not taken as seriously as a randomized controlled trial. Now, while this evidence hierarchy may be important in certain areas, I think we've outgrown this evidence hierarchy, particularly when you come into genomics and pharmacogenomics. And really, what we need to consider with the regulators is what kind of evidence will be required so they can undertake regulatory action to be able to start thinking about implementing pharmacogenomics. What kind of what do guideline makers need in terms of evidence to be able to change guidelines to implement pharmacogenomics? Also, how will regulators handle the fact that there are many people now starting to use artificial intelligence in terms of undertaking genomic interpretation of variants in the human genome? And I think this particular hierarchy is no longer useful in genomic medicine. We need to be able to use any kind of evidence that we have in an intelligent manner, synthesize all the evidence we have, and then take evidence-based decisions on what we require. The regulators will need to accept this kind of multiplicity of real-world data in order to be able to make changes to drug labels, to guidelines, and so on, so that we can actually take this forward into clinical practice. So as we move forward in the future, at the moment, we are particularly focusing on common variants in the human genome, typically more than 5% minor allele frequency. And as I said, very few have been implemented. So we do need to implement those, and that's a big focus at the moment. As we move more into the era of human genome sequencing, we will discover more and more rare variants, and a major challenge there will be that we often do not know the function of those rare variants, and we will need to develop ways of being able to really understand those rare variants and take them into account when we look at the pharmacogenetic variation. And at the moment, when I prescribe, I take into account other diseases the patient may have, other drugs they may have, and we you really use this in isolation without looking at the genetic variants. So in the future, we will need to take into account the common variants, the rare variants, as well as the disease factors, as well as other drugs that people are taking into multimodal algorithms, then to define what particular drugs patients require. That will make prescribing a very complex issue for most clinicians, because most clinicians will not be able to interpret the multiple amounts of data that's going to be thrown at them. And so what we really need are decision support systems. And what I mean by decision support systems are those which turn data, the data kind of data I've been talking to you about, into information and usable knowledge. And this usable knowledge then leads to wisdom and improves decision making. And here, in terms of prescribing, what we want to do is to utilize the data on diseases, on the genomic variants, turn it into usable knowledge so that the clinician can make a good prescribing decision that prescribes the right dose. It avoids drug interactions and improves the clinical outcomes of the patient. Unfortunately, decision support systems are still in the infancy. Some prescribing decision support systems have been developed, but they produce a lot of alerts, and when a doctor keeps seeing alerts coming up on his screen, he'll often switch them off, and that's got alert fatigue. We need to develop much more intelligent decision support systems, which not only reduce the amount of alert fatigue, but also include genetic information. Another challenge will be the fact that we use many different types of computer systems in our hospitals, and interoperability may also be an issue. So just to finish off, we're at the moment, we're at an iPhone 12 which I think has just been introduced. 50 years from now, if my mathematics is correct, will be on iPhone 37. At the moment, we have GPS on our iPhones or our phones, and that's for global positioning system. In 50 years' time, GPS will also mean genomic prescribing system. We'll probably be carrying our whole human genome sequence on our phone. It'll be linked to our electronic health records, our laboratory tests, all the sensors the iPhone will carry, plus other sensors we'll have, and it will incorporate AI algorithms in there, so that you will only have to present your phone to your doctor, who will then be able to make sure that you get the right drug at the right time, at the right dose, taking into account all genomic factors as well as all other factors. So, so this will lead to a different way of being able to treat patients. There will be much more patient empowerment, because patients will be carrying their own information, and patients will come to you saying, look, I've got this particular genetic variation, and how do I go about changing my treatment, or I need to have my treatment change. And that change in patient healthcare practitioner relationship is a really important part of the evolution of the practice of medicine, and genomic medicine is a catalyst for that change in relationship between the patient and the doctor. So I just want to thank the people who work with me and many of my funders and so on, and thank you very much for your attention. I'm happy to take any questions.
Great, thank you for that fascinating talk, Sir Munir. If anyone has any questions, please could you put them in the chat, and then I'll put them to Sir Munir. And while we're waiting for questions to come in, could you briefly talk about your role with the Commission on Human Medicines in the vaccine approval process?
Yeah, so the Commission on Human Medicines is an independent advisory committee for that advises the Medicines Healthcare Products Regulatory Agency on licensing decisions, and it has various subcommittees. And I was chairing, as I mentioned this morning, the committee which has been looking at the COVID vaccines. And when we look at any product that comes through to us, irrespective of the vaccine or drug, we look at it on the basis of three things: the efficacy, safety, and quality. And we need to take all of those into account to come to a decision on benefit risk of a particular medicine. And if the benefit exceeds the risk, we then can recommend, and we're an advisory committee, we recommend to the regulators, the MHRA, that this may be suitable for licensing or authorization for use, which is what we did with regard to the Pfizer-BioNTech COVID vaccine that was on the news this morning. Hope that helps.
Yeah, very interesting. Could you briefly tell us, I mean, if you can, any thoughts you have on the Moderna and the Oxford vaccine, and what you think the timescales for their approval might be?
So I can't, unfortunately, because that's commercially confidential, and obviously, so I haven't seen all the data and so on that's coming through on that. So it's difficult for me to answer that at the moment.
Okay, no problem. Let's move now to audience questions. So we have a question from Kwang Nguyen. Apologies if I'm butchering your name. And he asks about whether the definition of pharmacogenetics and transcription rates and the differing transcription of different genes effects is taken into account when thinking about prescribing based on someone's genome.
Yes, and that's a really important question, really important, very good question. And we at the moment, we don't really take that into account. And what, basically we are mostly looking at the exome, the exonic regions of the genome, and we don't really take into account any of the intronic variants, and which may be important as well in terms of pharmacogenomic variation. We don't take into account alternative splicing, etc. So there are many different aspects we still need to look at to take into account the overall variation of how the genome affects drug response and disease susceptibility as well. And as with disease genetics, people are starting to look at the kind of area such as intronic variation. And I think this is a big growth area for the next few years.
And if I might just add on to that question, do you foresee any challenges in the future with, we present, genetic data to provide it to everybody and people are able to kind of inform themselves of, oh, this is my genome, therefore this is the treatment I should get. But do you see any sort of problems with that moving forward, with almost too much information and, you know, people taking out too much, you know, it's like hypochondriacs and WebMD kind of thing?
Yes, absolutely. There is a danger of that. But that's why we will need some evidence. You know, we can't just say that here's a variation in your genome, that, you know, you need this particular drug based on theory. So we will need the evidence of utility as well, and that's going to be important. So you know, what you should do is to base the variation you have in your genome on evidence for utility, which makes a difference to patient care. And you know, if that information is then available to the individual to say, well, you have got this variation and there is good data that a particular drug may be better for you than another drug, then that's a good thing for the patient to be able to remind the physician about. I don't mind patients coming to me with their 23andMe result and saying, what does this mean for me? But because I have a background in pharmacogenetics, I don't mind that. But unfortunately, many people haven't been trained in that. And, you know, it's kind of a nightmare scenario to be able to get that kind of, information from a patient.
Great. Before we go on to the next pharmacogenetics question, I wondered would you be able to speak at all about the Pfizer-BioNTech vaccine and your role in looking at the data and approving that? Is that something you'd be able to discuss?
So as I said this morning, we worked with an expert group of people, immunologists, virologists, epidemiologists, toxicologists, pharmacologists, and we looked at all the data that was presented to the MHRA, including the raw data, and looking at those areas which again mentioned, quality, efficacy and safety, and really dissected out, pored over the data, deliberated over the data for many months, and you know, came to the conclusion based on the fact that the efficacy, as I said this morning, is 95%, and in terms of the side effects, those are relatively minor, as you'd expect with other vaccines, and usually last for 1 or 2 days. So the overwhelming benefit risk ratio is positive in terms of this particular vaccine.
And are there any concerns that new side effects or adverse effects might emerge as we start to vaccinate, you know, hundreds of thousands and millions of people, as opposed to only the tens of thousands that were vaccinated in the clinical trial?
So far about 20,000 have been vaccinated in that clinical trial, and you know, obviously rare adverse effects cannot be all detected within the 20,000 that have been vaccinated. And that's really important, as I said this morning, that there is a post-marketing surveillance of these vaccines as they are deployed. And the MHRA has got very good post-marketing surveillance activities, such as the yellow card system. They'll develop an active monitoring system where they'll invite people who've been vaccinated to join this active monitoring system, and the people can then report on this active monitoring system as to whether any problems are occurring. And so it is really important that people participate in a system like that.
Okay. And I have one final vaccine-related question, which is the European Medicines Agency has decided not to go ahead with emergency authorization for the vaccine, while the UK has. I wondered if you have any thoughts on why they might have chosen to spend more time and look at a larger fraction of the data, and why the UK has chosen to move more quickly.
So I don't do very much with the European Medicines Agency, and you know, I can't really answer for them. As I said this morning, and as did June Rain from the MHRA, that we took a very stringent approach. You know, there were no corners cut at all. You know, we worked tirelessly. The MHRA staff worked tirelessly. The expert working group had 40 hours at least of committee time, apart from the reading time outside that. And I spent weekends, evenings on Zoom with the expert committee, really looking at all of the data in a stringent manner. We felt that this was a public health emergency, and we wanted to be able to look at the data in a stringent manner so that we could reach the conclusion that we reached.
Okay, sounds very thorough. I think we'll have one final question on the pharmacogenetic side of things, which is from Anna. who asks, would the new solutions you talked about be mainly aimed at secondary and tertiary care, or will it also find a place in primary care? And if it will, how many years away do you think this is?
That is also a very important, very good question. So most of prescribing in the NHS and in many healthcare systems goes on in primary care. So really, for pharmacogenomics to succeed, it has to be embedded in primary care. The trial that I mentioned, UPGX, started off in secondary care in terms of the patients are, admitted to the trial when they're admitted to hospital, but they take away a card with them and they can present it to GP when they get a second drug prescribed. So in a way, what we are doing is transitioning from secondary to primary care. But I think in the future, what will happen is that GPs will need to be able to understand pharmacogenomics and be able to, using the NHS genetic test directory when the pharmacogenomic data is available on that, they will be able to then ask for the test themselves in primary care. Another model of pharmacogenomic testing in primary care will be through community pharmacy. Community pharmacies already provide a lot of different activities, testing, blood pressure, cholesterol testing, but in the future they may actually provide some pharmacogenetic testing as well. So that might be another way of implementing this into primary care. So I really do think that primary care is a hugely important area for pharmacogenetic testing, but it may take a while before it gets in there, given all the constraints in terms of the education, the decision support systems, etc. Hopefully those will be embedded in the next few years, and then that will help with regard to implementing pharmacogenomics into primary care.
Okay, very interesting. Would you have, I know you're extremely busy, but would you have time for one final question?
Sure.
So I think we'll go with, there's just been a new question that's come through. Let's go with that one, actually, from Kwang again. If you were to make an investment in a company targeting pharmacogenetics innovations, what would be some important considerations you might have?
Thank you for that. You're obviously thinking about investing in a pharmacogenetics company. So I would look at their technology that they have, what they're offering, and what is the evidence base for what they're offering. There are many pharmacogenetics companies there offering panels of testing for pharmacogenetic variants, and often you find that the evidence base for this pharmacogenetic variants is poor. And if you look at different companies offering different pharmacogenomic panels, you find that there's variation in the pharmacogenomic variants, including the panel, and that creates confusion, also creates lack of, reduction in confidence as well. So I think it's important to be able to look at it from that perspective.
And just as a follow-on to that, is the NHS looking to collaborate with private companies with delivering pharmacogenetic solutions in the coming years, or is it going to keep it more in-house?
Well, so the NHS is now the most advanced healthcare system in terms of genomics and mainstream implementation as a whole system, and that's largely related to the 100,000 Genomes Project. As part of the after the 100,000 Genomes Project, the NHS has developed the Genomic Medicine Service, and there are 7 genomics laboratory hubs now throughout England. This is for England I'm talking about, and the similar systems are ongoing in the developed nations as well. And those genomics laboratory hubs have the capabilities to be able to undertake the pharmacogenomic testing that I was talking about earlier. So if you have that in-house testing capabilities, the majority of them will be done within in-house, because you have all the sequencing platforms, we have all the genotyping platforms in-house, and the expertise to be able to interpret as well as report that data. Clearly, there may be some specific tests, that may require collaboration with industry. In which case, it's not doesn't mean that the NHS won't collaborate with industry, but it depends on what particular area it is, I would have thought.
Great. I think on that note, we'll finish up. Thank you so much, Sir Munir, for giving up your time to be with us today. You are exceptionally busy in this current global pandemic, and we really appreciate it. To everyone who's attended, thank you very much for coming. We are launching our nationwide bioentrepreneurship program More details can be found on our Facebook page, and the deadline for sign-ups is the 17th of December. So I'd really encourage you to go and have a look at that. And on that, yeah, thank you everyone for coming, and thank you again, Sir Munir.
Bye-bye.
Bye.